Okay, my friends, this is almost too good to be true. This is what you call a twofer or more. Now, why am I saying it's a twofer? This is Caltech, and they're going to be using ultrasound to move cells around in the body and build tissue. What I want to see them do is move the bacteria into the immune region of the cellular walls, and that is this area right here, this dark area. If they can push certain types of bacteria into this area here, that I believe is what creates your immune system, because bacteria create enzymes. Enzymes do all the chemistry, literally all the chemistry. Enzymes can create a chemical reaction instantaneously of hundreds of thousands of other molecules. Boop, instantly they all change within the speed of light, because it's a magnetic snap. One little particle comes up and goes bang, and they all change at once. So, and that happens in here. And there's new photography showing these little buggers running around through here. It's a fluid-filled highway. And this is brand new. This is not something that they knew about before. This is just being studied now. This, your newfound organ, your meature interstitium. This is from March of 2018. This is only a few years ago. And this kind of stuff works at a glacial pace, trust me. I have been following this very carefully. This is like the skin on your skin. Right? You have different thicknesses of skin. On your hands, it's just very, very tough and thick, and on your feet and so forth, elbows and that. On your face, totally different situation. But under every one of those layers of membrane, basically, which is your skin and your wrapping of your organs, the membranes around your organs, you will have this layer. It's 100% covers every organ in your body, your muscles, your urinary tract, your digestive system, your eyeballs, everything has one of these layers. That layer is there to say, you guys can stay out here, you can come in here and invade a little bit. I can deal with that, we can slough that stuff off, we don't care. But you come down here, we're not going to allow that. What you have to have here is the bacteria. Because if you don't have the bacteria living in there, you can't create the enzymes. You don't have the enzymes, they can't do the chemistry. You can't do the chemistry, the guys walk right through. They go right through your membrane and into here. That's what's called cancer. All right, when I just said cancer, this is what cancer is. It's invasion past this layer, basically, which is your membrane. Now, this is what cancer looks like in a little drawing. These are your membranes. The cancer cells growing through the normal tissue, which is your membrane, down into the vital flesh. This is supposed to stop. Well, the layer right here is supposed to stop it. It can come in here a little bit, but once it breaks through that layer that I showed you, that thick layer, then it's all over. Well, it's not all over. You should still be able to attack it back, because you're going to get cuts. You're going to go way deep into here. People get them all the time. And if you have the right enzymes, they will go down and attack the invaders that are already deep into your tissue. But if you don't have the right enzymes, they can't even attack them when they hit this layer. That's called immunocompromised. That means if you get hit by stuff, you can't defend yourself, you're going to get attacked. They're going to go right through. The cells have junctions. They're held together. They're called tight junctions. And if those junctions together, where each cell meets each other's cell, if they're not tight, the little bacteria, the enzymes or viruses, whatever you want to call them, are going to invade you. And once they get down here, they can take over your cells, because all they are is programs. They just reprogram your cells. That's all. And then your cells start working for them instead of for you. But you should have some other bacteria that will recognize those bad guys and go down and kill them. That's what an enzyme does. It's a killer molecule. And it is an elegant molecule. We're not talking any puppy molecule. We're talking about a really monster. molecule. Okay, so why do I say this is a twofer? He's talking about cells in the body, which is bacteria, enzymes, all of that biology. And then he's talking about using pulsed, energy to push them, which is, I also do that. This is what I use, light through the venturi, to create a similar type of an effect. All right, you got to see this. This is just too much. Listen to what he has to say. This is about how they're going to use certain types of energy to manipulate cells, to push them into the places where they should be for cell therapy. This is almost too much to believe, because this just came out 19 hours ago. I've been talking about this for a while. Now listen to this.
Actually trap and manipulate cells that we've put into the body as a cell therapy.
You see, can we actually trap and manipulate cells that we've put into the body as a cell therapy to help them get to the right place? That is the key. If you have the cells in the correct places, those cells will do their job correctly. But if they're outside of the mainstream, they can't get through these membranes. Now, these cells are extremely, extremely elegant. They're white blood cells and so forth. And inside those cells, they make chemistry. Now, that's why I say this is a 2 for 3 for 4 for, I don't know how many it's going to end up being. But we're talking about cells. We're talking about how the membranes work in the cells to keep things out or let things in. We're talking about different frequencies of vibration that create different openings possibly to allow these things in through, you'll see the research they're doing. It just exactly dovetails with what I'm doing. Now listen to what he has to say about pushing the cells into the right places. Listen to this.
To help them get to the right place. Right now, when you introduce them, they kind of go all over the place. And we think that this could potentially be a way to allow us to pinpoint or guide where they go inside the body and selectively only go to areas of disease, for example.
Let me just explain something. Every single type of molecule, every single type of biology, every single type of chemical has a magnetic signature. If you can tune exactly to that magnetic signature, you can pulse it by hitting it at exact correct frequency. This is what they're going to be talking about. You hit these molecules at the correct frequency, you can push them. And they're doing push to shove exactly what we're doing with our light research. Rod Warren's, the Warren effect, discovered how we can do this pushing, exactly like he's talking. Now watch this.
All right, so we want them to go into the tumor, and we want them to aggressively treat the tumor, and we want to be able to kind of tell them where to go and what to do, and this gives us a chance to literally hold their hand and tug them to a particular location. And so we're very excited and keen to try to do that.
This is Caltech. All right, so how do they tell the cells where to go? They're using what's called ultrasound. Where is it? Oh, yeah, here it is, ultrasound. Now, what is ultrasound? It's a certain frequency, which means how fast the pulsations are going, and they're using it to push molecules that have a certain resistance to that exact frequency. They'll say, no, you can't come through here with that frequency. He says, oh yes, we're coming through. And it just pushes that molecule to where it wants to go. Otherwise, they go right through. It's got to be exactly matched to the frequency that the molecule you want to move is opposed to. It's as simple as that. It says you can't come through if you're that long. That's it, you can't come through. It says, I'm coming through, I'm coming through, I'm coming through, I'm coming through. And it just keeps pushing it. That's what he's going to be talking about right now. Now we're going to go through this whole palette in a second, but this is exactly what we're doing by pushing particles apart, because we're creating these same patterns. And what we did was the same sort of thing they're doing. We pinpointed the light photons to push. These are pushers, and they will push and they'll keep separated from each other because they're magnetically separated. And I don't know if they picked that up or not in their research, but I'm going to certainly point it out if they didn't, or if they did, congratulations to them. I haven't looked at this deep enough to see what their conclusions were, but let's take a look at it now. Okay, so not only are we looking at the cellular biology and what its actual magnetic signature is. That's what I want to find out. What is the magnetic signature of these different types of cells? And we need to know what cells create what enzymes. Now, this is what they found out your cells look like. And this was a couple of years ago, 3 years ago. And again, everything goes so slow in the medical arena, and nobody wants to touch anything new because they're afraid of getting sued. I understand that, but this is what they found, and it needs to be deeply researched. This is GoCall. Okay, so this is from UC Berkeley. This is GoCall, and they had come up with a new technique of seeing into the cells in this extremely detailed look down in while the thing is still alive and doing the life processes, and they can watch the bacteria and the enzymes and the cellular layers and all of the fluids doing what they do. They discovered there's 2 types of bacteria. One goes out and sniffs around and finds out if there's any problems. That one alerts the other bacteria. It goes and gobbles up all the enzymes, goes out and delivers them and kills this, or goes to digest food, whatever it is. Whatever it's told to create the enzyme, it goes out and delivers it. Here's what he saw, and he freaked out. Listen, I still remember the moment. Listen.
Still remember the moment. It's something I will never forget. The hair on my hands just stood up. It's a microscopic universe within each cell.
This is an unprecedented view of the cellular world, where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing only on the crawling immune cells, we've noticed 2 classes of them. One team was not hungry at all, but it was very active in terms of trying to figure out, what the environment is. There was another one that was kind of slouching around with a lot of food in its belly. We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments in this crawling immune cell as it's kind of, scooping up its environment.
All right, look at the size of these cells. Remember before I showed you the size of those immune cells that were lining the membrane? That's where these bacteria live. And what it is that's an enzyme. Or, I'm sorry. These, I believe, are enzymes or polypeptides, peptide chain, little tiny bits of peptides. And what's going to happen is they're saying it's in the ear of a zebrafish. Well, everything has a membrane, so I don't care where it is, it's in the membrane. And this is the immune cells, they're white immune cells. And there's this main guy that runs around in here grabbing all the stuff, and he's going to squirt out through this membrane because he's allowed out. And he's going to take that stuff out and deliver it to do whatever he does with that enzyme, but it's a chemistry set. And here's what happens.
Unprecedented view of the cellular world, where we can...
You see, that is probably a bacteria. And I'm going to show you all these things I'll show you the stuff I have about this. It's going around grabbing up whatever this stuff is. Now, that could be, like I say, enzymes, polypeptides, whatever. But he's being told, go get this stuff and go out and deliver it for whatever reason. What are the reasons? One of them is you're being invaded. Go get some enzymes, go out and do your chemistry. Now, what is the chemistry? It goes around and it collects up all these what are called proteins. And when you see them, you're going to freak out. A protein is something as complicated as that, made by a bacteria. And there's thousands and thousands and thousands of different actual chemistry sets, because that's a chemistry set. Now, what does it do? This guy goes and grabs them. He collects them up. He goes out to deliver them. And what does he do when he delivers them? He goes to find the guy, the target that the other guy, the sniffer guy, said, here's the chain, and he'll come back with something like this, and he'll say, if you recognize this, if you can find that type of a chain, attack it. Go and attack it. That's what the other guy did, that guy that doesn't have any food in his belly.
Can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing only on the crawling immune cells.
Now listen to this
you
now.
notice two classes of them. One seemed was not.
This is the one that's not active. It's very active, but it doesn't eat anything. Listen to what he says.
Hungry at all, but it was very active in terms of trying to figure out, what the environment.
It's trying to figure out what's going on. It goes out and it says, whoa, there's a guy there that I don't like, and here's his chemical signature. It comes back to that big, huge cell, and it says, hey, here's what's going on. If you see this, make an enzyme to go out and kill it. And that's what the other one does, is make the enzyme, they squirt out through the cell walls and attack
There was
the
another
invader.
one that was kind of slouching around with a lot of food in its belly. We can actually.
He says a lot of food in his belly. What it is it's created an enzyme. That's a chemical package that goes out. Now what happens with this is this. You see that? That's what it's got to attack. And if there's no chemistry to do it, it can't attack it. It just keeps going through you, through the wall. Now, so let's say you do have the chemistry to attack it. The enzyme squirts out this product. The bacteria grab it all up, they go out into this layer here, and it attacks it. Now, how does it attack that? There might be 10,000 cells here. They all have the same chemical signature. They all have the same magnetic property because they're the same kind of cell. This one knows what that magnetic property is. One single enzyme, one single cell, which is an enzyme, can go out, and if it finds something like this, and it has the correct chemistry, it can go click, and every one of them dies instantly, because it breaks all their chemical bonds from the magnetism this smashes into it with. And guess what? This one doesn't even care about it. It just goes on and sees if it can find any more of these bad guys. And it breaks them down, breaks them down, breaks them down, breaks them down, one at a time. That's what an enzyme does. It doesn't even get used up. When it breaks 1,000,000 of these, it like just bam, they all are dead instantly. Well, let's say most of them. And this one doesn't even care. It goes on. It doesn't even get used up. Now, what do they look like? How elegant are they? Well, they're the most elegant thing you've ever seen in your life. This is the most rudimentary look at these are the triggers, the sheaths, the proteins, the enzymes. They're all are enzymes, proteins. Yes, they absolutely are. And they're not anything cheap like that. Here's what a real enzyme looks like. Boom. You see that? That's just like this, only on steroids. Every one of these little spots is what's called a dipole moment. It's a magnetic spot that's exactly in the correct position, and it's exactly a certain polarity, and it's exactly attached to other molecules, and it's exactly like this. It will never change if it's capable of doing its job. Once it becomes denatured, overheated, or damaged by something, blip, that goes, and it's done. No worky anymore. No worky. But if it's like that, it goes out and does its job. That's how elegant the chemistry is. And how does this get made? It gets made by a bacteria. I'm not kidding you. All right. What you're going to remember now is that there is more bacteria in you than there is you in you. There's more bacteria cells in you. They're single cells. One cell. Single cells, but they have programs in them. And what is the program? It's the nucleoid DNA program. All it is a bunch of this stuff, all twisted up in knots and so forth, and twisted in a double helix and so forth. Now, they all have these well, not all of them, but I believe most of them have flagella. Some of them have one, some of them have 10, some of them have who knows how many. Some of them have fur all over them. Some of them are completely smooth. It depends on where they're going, what they're going to encounter, what they have to stick to, how fast they have to move, what program is in here. What is that going to do? Is the program in here to go digest food? Is the program in here to go kill COVID? Is the program in here to go kill some kind of other invader? Okay, a little interruption there, but I'm going to just, I can't remember what it was, but I'm going to just explain to you each one of these little components. What is a ribosome? Well, a ribosome is actually like a little program, basically. And what this does, it's a molecule that binds messenger RNA and transfer RNA to make the enzymes. These are the little production packages. This is the DNA of this particular style of bacteria. That bacteria has to have that DNA in order to make these ribosomes that program the cells. They go out and program other cells to do a job now, or make enzymes. Ribosomes actually make the enzymes. So a ribosome is an RNA. It's not boy, this is tough to understand, but I'm just going to try to cut to the chase. Ribosomes make copies of themselves. Think of it that way. They are the manufacturing factory inside here that makes copies, and those copies make enzymes. Those enzymes are the products that are gone out and delivered to either break down food or to attack other things or to create chemistry that is good for your body. You need to produce something to bring over to keep your bones in good shape. It'll go out and deliver that, some kind of connective tissue, collagen or whatever. But that's what these things do. Inside here in the ribosome is what this is. Think of it this way. Now, what is it squirts out of here whatever this program is. So let's see how it squirts it out. Let's see. Here's how it squirts out. Whoops, that's not, well, this is what it squirts out, is these. This is the messenger RNA and transfer RNA. And see, this is a messenger RNA, and these are the transfer RNAs, and this is what does the programming. As this thing streams by. And it has 20 different codes, which are the amino acids. And they come down the line and it programs. And if it's not in that exact sequence, it won't work. And after that sequence gets to the end of its snip and it snips off, it goes and it creates an enzyme, its enzyme package. And that's right here. Well, this is what it ends up being. I mean, these things are just absolutely stunning when you look at what it can create. And this is how it does it. Coming out of here, this is DNA that's in here. It says, I am a certain type of bacteria. I am a lactobacillus Scooby-Dooby. And every time I send out my codes, they're going to come out just like this. And then it's going to go chunk, done. And at that point, it's programmed into some RNA and DNA and all that different stuff, messenger RNA, transfer RNA. Now, and remember I talked about the ribosomes? This is where it gets programmed. So it comes out as a chain of the correct proteins dependent upon what type of a bacteria it is. It's all dependent upon the bacteria. If you don't have the bacteria, I don't care what you do, you're not going to get the right codes. Now you may get something pretty close, and it might work. That's the problem. If you don't have the exact right bacteria. Now, can we push these bacteria to where they're supposed to live. Because if they're living there, they can self-produce, they can reproduce themselves. If they're living and they're healthy and they're getting the nutrition they need, and if somehow they die off because of antibiotics or whatever, and we can take some more of that exact same bacteria and push it right into where it's supposed to live in these white blood cells, that's a pretty good outcome. Can it be done? I don't know. That's what they're trying to do. All right, so don't forget, this is what they're trying to do, is to push these cells into the correct places at the correct time to do the job they want to do. Now let's see what he has to say right from the beginning here. All right, they're talking about plants and growing and how the plants rise to the top because they have certain bacteria to create pockets of air so that they can get up to the sunlight. Now listen to what he has to say.
That originally evolved to help cyanobacteria that live in water rise to the top of the water so they can get enough sunlight. So there are these little air-filled proteins that self-assemble inside of these cells.
Now see, he's saying self-assemble. No, they don't self-assemble. There has to be some program to assemble them. And then there's got to be something that assembles them. So you need the program and then you need the assembler. And those are both bacterial functions.
To help them float.
A couple years ago, Mikhail had discovered that these structures, in addition to their uses naturally as a buoyancy device, can scatter sound waves and be
All right, they scatter sound waves because they are nothing more than molecules with a magnetic field around them. When you bounce a certain sound wave off them, it bounces off them. That's why reverberation happens. Now listen to what he says. So they can scatter sound waves. Let me come back. I know I'm going to keep cutting in on this, but you have to understand what he's saying. He said a couple of years ago they discovered these structures, in addition to their use naturally as a buoyancy device, can scatter sound waves. So they're just like hitting a balloon and it's bouncing back. That's all it is. That's the Kashmir effect.
naturally as a buoyancy device, can scatter sound waves and be imaged by ultrasound. And this
All right, when they say imaged by ultrasound, imaging means they can see how hard that thing bounces back, and they can say, ooh, that's a certain frequency. Let's look at that exact frequency and image that frequency.
opened up the entire field of research that's now called biomolecular ultrasound that allows researchers to connect the production of these gas vesicles to what's happening inside a cell.
We've done a lot of work, we're imaging that, and now with this work, we're trying to see if we can not only see where the cells are, but also tell them where to go.
All right, now here's where it comes in. As to these cells, they have to know their magnetic signature. They have to know their exact resistance. Let's think of it that way. If they know its exact resistance and they can hit it with exactly that frequency that moves it, they could very well do what they're talking about doing. Now this is what we work on. Rod Warren discovered that we could separate the particles of light, and as I'll show you, or have shown you, and I'll show you again right now, this is what happens when you push and shove light. You can separate them into bars, because the white ones want to push the other white ones away, and they'll say, I'll stay here and we'll create a buffer zone between us. You see the black buffer zone between there? It depends on how this is done, but they're going to do the same sort of thing, only they're going to do it with sound. We're doing it with light, which is like on steroids. They're doing it with sound. Now let's go back to see how they did it. All right, sound is ultra low frequency. You can't see it. It doesn't light anything up that you can visually see, but it does push and shove other particles back and forth. Now they're watching in this very, very low range with some special instruments, so they can actually see these pushing and shoving interactions. Now watch again, Caltech, here they go.
Ultrasound is a wave. It's basically a wave of pressure that's moving through a medium. So you have compression and rarefaction, compression and rarefaction.
All right, that's exactly what we have in our particles, compression and then a black spot in between, and here it is right here. This is exactly what we have. There's the compression, rarefaction, compression, rarefaction, compression, rarefaction, compression, rarefaction, compression, rarefaction, explosion. That's because we put it through the Venturi. Now they're just trying to push the particles. We're trying to divide them, which we did. We did divide them, and here it is divided right there. The black cannot get through. The black is a fixed particle. It cannot change size. The slit here is so small, and the particles of white can slide right through, and the black just forces them through like bowling balls. Boom, boom, boom. And all the pins have to go through, and only the white pins can go through. The black balls bounce back out this way, unlike at a bowling alley. They just can't get through. They just keep piling up here and slam and slam and slam and slam and slam. So we should be able to harvest this in pulsating energy down through a diode, a photodiode, some receiver transition metal collector, which is all the different transition metals on here, thin film, suck all those electrons down through a diode. Once they get past, push, 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 they can't come back. So they pumped into here like blowing up a balloon of electricity. And we could use it for anything you want. And this could be in your, like a little handheld device. With any kind of power you want. This is so much power, you should almost not even be able to use it, is what they say. This should be at least 200 times what we started with from here. If they're correct about their calculations of the energetic values, and I believe they are, because luminosity, how bright something gets, is the key to its power. That didn't have any brightness at all until we hit this is like an atomic explosion. And all we did was restrict it with something similar to this. It's not exactly like that. But it's a Venturi. All this is a Venturi. It's nothing earth-shattering. So can we push particles using these little white pushers? Because the white lines are the pusher. We can actually focus it. See this? That's how focused we can get that white particle beam. That's just stunning, the amount of focus. It's outrageous, to be perfectly honest with you. And that's fission right there. And that's fusion when the particles come back together. They were stuck together, the black and the white, no question whatsoever. So we got a lot of thinking to do just in this particular side of the physics of light. Now we have to start to understand the interaction of the enzymes with the layer of tissue, which is like, I believe this is like facial tissue. See how thin the surface is here? Or it could be some organ, I don't know. But I believe this little red spot layer here is the layer that has the white blood cells. And once they get through this layer, there's still some down here that look like they're capable of fighting back, but primarily you're going to have one layer of membrane, which is, I think I showed it here before somewhere. All right, this is the primary defense borderline. I'm sure you have these other ones. See, there's probably one there. They're floating around here and there, I'm sure. But this is the main borderline. Do not come through here or we're going to be in trouble. And these are the big big white blood cells. And inside those cells live all that stuff that Gokul was talking about, showing these bacteria running around in there, and they jump into the fluid-filled highway, and they run over to the tissue type that's got to be attacked, or food that's got to be dissolved, or whatever it is. Don't forget, you have more bacteria in you, of cells of bacteria, than the cells of your human body. This is no joke. This is the stuff that makes your body work. If your bacteria is not there, your body's not going to work. Enzymes are the product of the factory, which is the enzyme. The enzyme, the protein, is the factory. That protein right there comes out of the bacteria, which is the factory. I know I just screwed that up, didn't I? Let's start with the bacteria. The bacteria squirt out all of the proteins, and the proteins are the enzymes. Proteins and enzymes are the same thing. Enzymes are proteins, and they're these elegant molecules. So if you don't have the bacteria, you don't get these things. Let's just put it that way. You do have a program built in you, and they always point to that, and they say, oh, you got a genetic problem, this is mutation, da 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 da. No, not necessarily. If these bacteria are not here to create these elegant molecules, you're going to be sick. That's why some people are sick and other people aren't. And almost every single time, they have some kind of a gastrointestinal issue, which means that the bacteria that live in your gut, this is where all this stuff happens, is in your gut, primarily. And then they roam around through that fluid-filled highway. But they're almost exclusively in amongst the gut tissues. That's just their habitat. That's where they live. And they go out and attack food when it comes down. Most of the stuff comes through your throat and your lungs and your stomach. So they have to be tons of them in there. And you have to have them in your heart, your lungs. Well, they're everywhere. But they're specific to different places. In your lungs, you have to have specific ones to keep the junctions tight. You have to have tight junctions between your cells. In your lungs, if they start to get loose, your collagens and connective tissues are not bound tightly, you get invaded. They go right through there. It's called tight junctions. All right, now let's go back to see what he has to say. Now don't forget, they're using sound, ultrasound. It's like certain frequencies, which are wavelengths. A frequency is nothing more than this. All right, a frequency is nothing more than this. When you look at it, it looks like a wave like this, but it's actually spinning. These particles spin, and I can guarantee you they spin because I can show you them spinning. But when you look at it from the side, you see a wave. And the longer the wave, the more shifted towards the sound. And when they're real long, and they're just like way long like this, that's when you're in sound. That's bouncing back at me. Light comes back much faster and it glows into my eyes. Sound does not glow my eyes. My eyes won't glow from that. I can't see a glow. I can hear it because my ears are tuned to that very low frequency, very long frequency. My eyes are tuned down to frequencies like this. Now, I hope that gives you an idea of what we're talking about, the bacteria, how it's assembled. The bacteria makes these little polypeptide chains. They come in here, they get programmed, they go out, they attack, they do all the things they need to do, working through these bacteria. And the bacteria, I believe, gobble them all up, and with these little flagellums, they turn 100,000 RPM, some of these, 100,000. They can go through your fluid-filled highways and instantly, literally almost instantly. I'm not kidding you. It's unbelievable. And that fluid-filled highway has just been found, called the interstitium. And this is what it really looks like, really up close and personal. It's down in through here, these fluid-filled highways, and that's where all these little balls are. And this is something they just now found a couple of years ago. So this needs to be researched closely, and this is what it has to do, is come through that layer and attack these cancer cells. And the only way you can do it with this elegant chemistry, and that elegant chemistry is only produced by these bacteria. And the bacteria create the enzymes, which is the elegant chemistry. Don't have the bacteria, you ain't got no elegant chemistry. Gabish? Okay, so they need to push the cells where they need to have them go. Now listen to this. Here we go again. This is Caltech.
Or imaging that. And now with this work, we're trying to see if we can not only see where the cells are, but also tell them where to go. Ultrasound is a wave. It's basically a wave of pressure that's moving through a medium. So you have compression and rarefaction, compression and rarefaction, compression and rarefaction.
That's what I call push to shove, push to shove, push to, and that one, this one pushes, and the other ones have white particles too. They push back. So it's pushing, this is shoving back. So you get a blank spot between all of these different layers. It's exactly what he's saying, exactly identical. So they want to create an acoustic pattern which will be an interaction of that exact frequency.
And when we create acoustic patterns, we're causing areas of compression to be over here and areas of rarefaction to be over here. So you have a higher pressure and a lower pressure zone. And we've shown that with this work, gas vesicles would move towards areas of high pressure, whereas normally cells tend to move towards areas of low pressure.
You see this? You see this? You see this? You see this? This is light spinning, literally spinning through this medium. And the light is on the outside of the spin. So the dark stuff is in the middle, and it's coming up and compressing at, you know, if you could see it, it would actually be here and over here on the other side of this tube. It's not a standing wave that's going like this. It's a spinning. It's spinning up and hitting there. And over here, it's still spinning. Now it's hitting down here. And now it's hitting back up here. That's all it is. And this is ultrasound. It's a sound. And like I say, this is coming through a tube. If you could see, there'd be a white coming right down here. If you could see all inside of it, it would be just like this. But that's because it's spinning, not because it's flapping up and down. All right, so here we go.
In those videos, we are generating an acoustic field, and the field pattern looks like a standing wave. So there's sheets of high pressure and sheets of low pressure. And
that is just standard for pushing and shoving of magnetic fields. That's just the way it works. I'll show you some magnetic fields that there's no way to deny that.
what you're seeing are cells moving to these areas of high pressure. And now when we change the frequency of the sound, the cells will move in accordance. So we're playing different sounds, and the cells are in effect dancing to different frequencies.
There's a lot of interest in engineering tissues for replacing organs or for creating living materials where you want to arrange cells into specific patterns. Now, with the help of ultrasound, we can get these cells to arrange themselves in the pattern that we want.
Now, what they're doing is a little more intense than what I was looking to do, which is to what's called engrafting, which means you have to get your cells, your bacteria, to live back in that area, into that interstitium area right here. And this is it's a membrane. It doesn't want anything coming in there that it doesn't know. So when these immune cells that you're putting into your body want to try to start living in there, they're probably going to say, no, you can't get in here. And it's just, well, if we live there, we can get along good, we can fight all these guys, we can be buddies, and so, no, you're not coming in here. How do you get them in there? How do you get them in living in there? How do you make them start to live in amongst all these other guys and get along? That's the key, engraftment. And when you do get new immune cells through, there's a process they call it the microbiome, replacing these, apparently most of the time you do get some sickness for the first day or two because your body is trying to fight back against these invaders, and they are invaders. And then it sort of gets used to them, apparently. And they start to sort of work together, but I don't think they get engrafted in here. It appears to take a while to get them engrafted. Now, could we push them in with this ultrasound? Maybe. Okay, this is something that's critical you understand. There's a lot of things that you have to understand the basis, and then you can understand the complexity. Now, what are we looking at here? Visible light. That's all the different colors we can see. That's light that bounces back at us. And our receiving hardware, our eyeballs, allow us to see these colors. And guess what? That is only this little region right here. All of these colors are in that little tiny region of wavelengths of particles. All right, and visible light, like I said, down here it's like here. Bzzz. Over the red it's waves over here. And in between it's all different things. And the fastest it's spinning is down here, and the most impact it has. This can hurt you, this can penetrate you, because it's spinning so fast, like a drill. And this over here is just gonna, almost nothing. Now, above or below that actually, the infrared rays, those are heat, all right? Radar is, radar. They can go and bounce back signals. FM radio, TV, shortwave, AM radio. These are all different wavelengths until they're very, very, very long. In here, there's a certain distance that our eyeballs can pick up with the lenses we have. And this is it right here, and it converts it into colors. Simple as that. Above here, you start to get dangerous. Ultraviolet, X-rays, gamma rays, they can penetrate into you and go right in through our basic protection. So you don't want to be exposed to these in large quantities. The same thing with X-rays. They do them, but you don't want that a lot. But this is what we can see. And this is nothing more than how fast these things are bumping into each other with this frequency. Now, I always thought they were always spinning. And I still think that is the case. Anything that's light, I believe, is a spinner. But then you get into infrared and radar and, are these bumpers? Do they just bump and the particles don't move? These particles I know move. I can tell you, no question whatsoever, because I can see them move. And I can see there are particles. Once you get down into sound frequencies, and maybe even here, they may not actually move through the air, the particles. They just may bounce. There's going to be some break down here to where you get to the point of just sound radiation or just banging radiation. Vibration. Let's call it vibration. You see this? The speed of sound in the air. Well, you have to take into account the temperature. All right, you got to take into account what the speed of sound really is. It appears to me that it's banging particle to particle, and it's not actually a particle moving through the air. It's a field against a field against a field against a field against a field. So the low energy particles in the sound range, and probably infrared and lower, are just bumpers. Nothing really moves. It just pushes me and I push you and I push you, and eventually it just tapers off. Light is a little different. Light is an actual particle moving. So don't forget now, speed of sound is determined by a lot of factors, and it's just how dense the air is in front of it. Boom, 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 boom. And how many particles it's pushing at a time. I'm sure some of them are going to sneak through, and some of them are going to bang into other ones. So that means, what is the density of what we're moving through? Can I move around things easily, or do I have to bang through them? All kinds of things. But that's what determines how far the sound is going to move through the air, or through the gases, or through the fluids, or whatever it is. You see this? I mentioned tight junctions. This is what tight junctions are. A barrier function of a lung epithelium depends on so-called tight junctions. These heterometric protein complexes, proteins again, are made from enzymes, form the sealing interface between adjacent epithelial cells. They're all packaged together, one right up against the other. Damage of tight junctions is the major cause of epithelial barrier breakdown during lung inflammation. tight junctions. It's all related to having the right chemistry, and these are the kind of things that make you easily invadable. Okay, I just had an idea. We want to be able to force these particles in, which whatever they are, the enzymes or the bacteria, whatever it is we need to get into that immune layer to have it take up residence there. If we could determine what the frequency was, that's good. But to get it through that barrier, how are we going to do that? There may be able to just be an injection we could do when it was being done in a local spot. Because once it's into the interstitium, if you can get enough of it pushed in there, it should be able to float through the system. So if you could put some collagen, to open up the, something to fight the collagen in a certain area so you could break through that with this pulsation of pushing that particular particle into that particular membrane, right in a very selective area, and then let it go through the whole body. And I know it will do that because when they put in tattoos, sometimes they get them into the interstitium layer, and they can actually find those particles on your liver and your lungs and everywhere. It's a certain chemistry, I forget what it was. I was working with some people in Germany, and they told me about this years ago. They said there's something going on with this whole layer. And I said, well, I see it everywhere. I said, but I don't know. And they said, the only thing we can determine was it must be some fluid-filled highway, because they found these tattoo chemicals in some of the fascia particles that they were, doing analysis on deep inside the body, not on your skin where your tattoo was. So they thought that was kind of interesting, and I did too. I said, "Wow, it was very interesting." That was years ago, and since then, they have actually called this a fluid-filled highway, a new organ. I think I showed you this, but this is it right here. And I'm telling you, this is the place right there. That's the one, and that's meet your new organ. This is only 2018. All right, so if we could push into that layer with whatever it is that we need to get in here, and maybe even open it up a little bit with some kind of a collagen breaker that I'm sure there's some chemistry, some enzymes that sort of break down the collagen, and you might even be able to use like a tattoo needle or something, whatever, but you got to get it in there. And then you got to make it take up residence in there so that it's not destroyed. It'll probably be fought back at for a while. And you're probably going to be not feeling well for a day or two. That's what I've seen so far. That's what the research indicates. There's going to be some reaction. Now, eventually, though, it should take up residence in there, and then everybody's happy and everybody works alongside of each other, and some of your issues hopefully get addressed because now you have the chemistry there. Now, what I was just looking at was how they use ultrasound in the body, and this thing here is nothing more than ultrasound. You know what this does? This tells you how far and deep in your stud sensor is in the walls. So when you run this through your walls, it tells you how far, if there's a stud behind there. So it's putting in a vibration, and it picks up a certain vibration back. It says that stud is over here and so forth. And then they have another one on this side. It picks up electricity when you're near an electric outlet. That thing goes off. But what it's picking up is pulsations of vibration. When you put it up against a wall, you're putting a vibration out and seeing if you can pick up a vibration back. And if it changes rapidly, you just changed somewhere in the wall. You went into some other kind of a chemistry or density behind where that vibration is going. Now it's coming back quicker. Now, if you pass over an electrical area, it'll start to vibrate into this with the field, because the fields are 60 cycles. You know, you can't put this on a battery or a magnet or something like that and have it turn on. All right, it won't do that, but it will do it with electricity. It won't do it on this stuff, but it will do it with an electric outlet. So anyway, what these do is there's field sensors. One of them puts out a field and senses it back. The other one just looks for the field. But they're both the same, basically the same. This does nothing more than a temperature reading. And when you point something at, like the screen here, 86 degrees, 85 degrees, this thing here, 71 degrees, that over there, 72, 73, magnets, 72. Temperature in here is probably about 72. Caesar over there, 72.7, 72.8. Holy smoke, 73. Caesar, you're running a fever, man. Anyway, this is what the energy relates to, is frequency, pulsation, absorption, and then you get into your transition metals. That's where we need to be able to understand how these transition metals absorb all these different frequencies. And that also is going to relate to pushing and shoving, because the transition metals, we may even be able to coat. Wow. Holy smokes, let me show you something. This is eureka moment. All right, I just had a really kind of interesting thought. If you had a certain package that you wanted to deliver, and you could target it right next to the tissue you wanted to force it into, and you could open up the junctions of that tissue, and you could coat that package with one of these transition metals that is easily understood. It's almost like a capsule of a pill. All right, you'd coat it with that, which you could easily target with an exact frequency. You say, the vanadium, it would be like hitting them and drive it right in. Or chromium, and drive it right in. It's an interesting thought. I hadn't considered that before. That was just a moment of inspiration. Okay, something I just learned. I didn't realize about sound. I knew sound was a wave, basically like the light waves. But in a light wave, I know there's no question it's a particle, and that particle is moving. That particle is 100% for certain it's a particle, and 100% certain it's moving. And I see it not only in the green, we see it in the red, we see it in the blue. It's a particle, it is moving, and that particle can separate into its component parts, which is the white and the black. Here it is right there. They separate off, the white and the black. And this is not unusual now. I thought it would just be almost impossible for this to happen, but it's not even that unusual. The white separates from the black fairly easily, but they really want to be back together, and that's the white is the energetic portion. Now, not only can we see there's a particle, now we go into the vibration. Totally different situation. First of all, we had particles of just like one or two of these little particles, zip, that's light, going forward. These are going bang, and then this one takes off, and it bangs the next one. So this one ends up being just about where it was, and this one bangs the next one, and it bang, 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 and that's all it's doing is banging the next particle. So it's sending out a wave of concussion, and that's because of the Kashmir effect. It tries to hit this other guy's field, and it says, well, no, and it bangs it away. Well, this one has to get somewhere too, and it's going to bang into this guy's field. So it's going to bang that guy, and it's going to be bang, 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 right down the line. That's not a particle going flying through the atmosphere. It's a particle pushing another particle, pushing, 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 until it settles down. That's what vibration is. Okay, so back to Caltech. The main point was, can we literally push these particles of whatever they are into wherever we want to push them? Now listen what he has to say.
Now, with this work, we're trying to see if we can not only see where the cells are, but also tell them where to go. Ultrasound.
All right, and they're going to use these little bumpy waves and force them to move into wherever they want them to move into. Now don't forget, if we can use transition metals, which is something that we can actually, use as a capsule, we could focus in an exact frequency to push that capsule where we want it. Of course, it'd have to decay somehow. I don't know. There's a little work to do here. It's nothing, finished up by any means. But there's going to be some way of defining what that particle is that you want to move. Some magnetic signature, some coating of metal, some chemistry here that makes it unique. Let's go with that. Once you can find the uniqueness, you can create a frequency that you can see. It'll just move it. And then if you can get it close enough to where you want to deliver it, and you can open up the tight junctions that normally would prevent anything from coming in, maybe you can push it right in there. Now, will it be accepted? I don't know. These are studies that should be done. I mean, I hate to see animal studies done, but that's basically the only way you'd ever be able to tell. Is it going to be something that's going to be infectious and hostile to the body, or is it going to be something that would say, "Ooh, yeah, you should have been here all along. What happened to you? Come on in." And that might happen. But I can tell you one thing right now. There's a lot of people who have very, very weak immune systems. And there's a whole variety of weaknesses. One of them is you can't fight off bacteria that's trying to invade you. One of them is, and because it's always continuously you're being invaded and you're stopping the invasion. That's what everybody's body is. Almost everybody has a lot of different diseases in them, but you have your opposite attackers. So they stay in homeostasis, they call it, equal but opposition, and they fight each other to a standoff, basically. That's about what it is. So you have to have the bacteria to do your job. Now, that's one thing, fighting off invaders and keeping an even keel of the ones that want to destroy your stuff. Because think of it this way. Every cell in your body is going to die, and they die regularly. They don't live forever. Now, they have to be consumed. There's a little trigger that says, hey, that guy's dead. But it's your own body. It's going to go over there and eat it. Now, you don't want it eating something that's alive. So you have to have the right bacteria, the right enzymes, the right sniffers, all of that stuff to detect what to do. Now, you may have dead tissue in you that should be consumed and taken out of there, and your enzymes are not working, so your bacteria didn't sniff it, first of all. And secondly, your enzymes aren't working to go clean it up. Because mostly they would say, oh, that's a genetic defect. It's not turning on the correct, receptor sites and blah, 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 blah. Well, nothing happens in you that's chemical, basically without a protein, without an enzyme, without a bacteria. That's the key. They are the chemists of life, bacteria.